Welcome back to MCB 170, Society and the Brain. This is lecture 13 that corresponds to part 1 of module 7. The title of module 7 is Becoming Us. In module 7, we will discuss brain development. A large part of brain development occurs during embryonic development, but the brain continues to develop after birth. The duration of postnatal brain development depends on the species, but in humans it lasts a very long time. The brains of humans continue to develop structurally and functionally until the late 20s, until age 27 or 28 on average. That's a long time. This means that the brains of most of the students taking this course are still very much in the process of developing. The first brain functions to develop are sensory and motor or movement ability. Then in humans, language develops, and then the ability to control oneself develops, and the ability to act according to moral values and ethical principles. What is common in postnatal brain development in all these systems is that they require exposure to the environment. The development of the sensory systems requires exposure to the sensory world. The development of movement control requires exposure to and interaction with the physical environment. The development of language requires immersion in a verbal environment. And we can speculate and say that the development of moral reasoning requires exposure to a moral and ethical environment. We'll talk about language and acquisition of social abilities more in part 2. In part 1, I'd like to focus on the development of the visual system because it is the best understood. Here's a couple of images of the visual environment. Here's a more or less natural image. It's not completely natural because there's a road and a wall and cleared fields, but mostly natural. And this is a completely man-made artwork. In both cases, we can ask, what is the basic visual feature? It turns out that the basic visual feature are bars of various orientations, little bars of light, like contour bars of different orientations. You can imagine that you could build up, say, the border of the road and the dividing line and the wall and the trees from a bunch of bars, little bars of light of different orientations. In fact, the bars that are perceived are rounded, so we think of them as elongated blobs. But you can think of them as elongated blobs or as bars from which the visual percept is built up. We'll talk about that more later in this lecture. The visual system starts with the eye, and here's a cross-section of a pretty blue eye. The iris is blue. Light enters the eye through the cornea and is focused by the lens onto the retina. The retina covers most of the back of the eye. The retina contains the photoreceptors, the rods and cones. The macula is a special part of the retina that has a very, very high density of rods and cones. As you probably learned in high school, the rods and cones are the photoreceptors. Rods and cones literally respond to photons of light. They'll respond to photons of light. And they kind of act like little neurons in that they can secrete neurotransmitter onto the next cells down the chain in the visual system. It turns out that the first 3 steps of transmission in the visual system occur in the retina. And the retina actually has 5 layers. Let's talk about those. The first layer in the retina are the photoreceptors, the rods and cones. The rods detect light that's black and white. The cones detect color. The rods and cones project to bipolar cells. The bipolar cells project to retinal ganglion cells, and the ganglion cells have very, very long axons. They project their axons down the optic nerve into the brain. But there are other cells involved also. Right between the photoreceptors and the bipolar cells are horizontal cells, and between the bipolar cells and the ganglion cells are amacrine cells. Amacrine cells. So altogether, the retina has 5 layers of neural cells. Photoreceptors, rods and cones, horizontal cells, bipolar cells, amacrine cells, and ganglion cells. And already, these 5 layers of cells are doing a lot of processing of visual input. In fact, they are detecting spots of light, circumscribed spots of light. And the ganglion cells will send that information, those circumscribed spots of light, back into the brain. So here we see the bottom of the human brain in diagrammatic form. The 2 eyes and these big optic nerves that go back into the brain and cross over in the optic chiasm, and then go to the lateral geniculate nucleus. The lateral geniculate nuclei are big subnuclei of the thalamus, and they're the main relay station in the visual system. Lateral geniculate neurons then project back into the visual cortex. And they project all the way back to the back of the brain, all the way back to the back of the occipital lobe. This is the first part of visual cortex, called V1. In V1, your brain will detect those little elongated blobs of different orientations. V1 is going to compute the basic visual percept, these little elongated oriented bars or blobs. As you move away from V1 into V2, V4, and higher levels of the visual system up into the temporal lobes, the visual features that are extracted become more and more sophisticated. In V1, you have oriented bars or oriented elongated blobs. In V2, you have corners. In V4, simple shapes. As you progress into temporal lobe, more complex shapes. And finally, up around here, we're perceiving really complicated things like faces. Remember, the fusiform face area is about here in the temporal lobe. What's happening at the very tip of the temporal lobe? Very sophisticated forms of visual processing that neurobiologists are still investigating. Now, what you notice immediately in this diagram is the crossing over of the projections from the 2 eyes. It turns out that the left eye and the right eye send information from the outside of their retina to the same side of the brain, but they send signals from the inside of their retina to the opposite side of the brain. What's going on here? Well, think of it this way. An image occurring in the right part of the visual field is going to send light to the outside of the left eye and the inside of the right eye. The outside of the left eye projects straight back to the left side of the brain. The inside of the right eye crosses over to the other side, to the left side. So input from the right side of the visual world is going to the left side of the brain. Similarly, input from the left side of the visual world is going to the right side of the brain. Importantly, and you can see here, input from the left eye is shown on the diagram as dashed lines. Input from the right eye is shown on the diagram as solid lines. You can see that both sides of the brain are getting input from both eyes. And that's very important for stereopsis, the ability to see in depth, the ability for you to just look into the world and see which objects are in front of other objects, to see which objects are close to you and which objects are further away. How does this happen? It's because the 2 eyes see the world from slightly different angles. And the brain can use the disparity, the difference between the images from the 2 eyes to actually compute the distance from the viewer of various objects. So binocular vision, depth perception, stereopsis requires the commingling in the cortex of input from the 2 eyes. We know a lot about the development of the visual system and probably about development postnatal development in general by studying this projection from the lateral geniculate nucleus onto the visual cortex and how the input from the two eyes is organized in the visual cortex. And the main technique used to study this is called autoradiography. Autoradiography is a histological technique. What is histology? It's the study of the microscopic structure of biological tissues. Autoradiography is based on radioactivity. Ordinary molecules like amino acids or hormones can be made radioactive. We already talked about radioactive forms of estrogen. They were injected into a mouse. They bound in the brain to estrogen receptors in the mouse brain. And after the mouse was sacrificed and its brain removed and cut into thin slices, so its microscopic structure could be examined under a microscope or with special techniques, researchers could find the places in the brain where that radioactive estrogen bound. And we found there was an awful lot of radioactive estrogen in the hypothalamus, as you recall from previous lectures. We're going to talk about a similar technique here in the context of the visual system. These molecules, like amino acids and hormones, can be made radioactive by substituting some of their hydrogen atoms for tritium. Hydrogen, as you know, is the simplest atom. 1 electron, 1 proton. The nucleus of a hydrogen atom consists only of 1 proton. Tritium is an isotope of hydrogen. Tritium has 1 electron, and it has 1 proton in the nucleus and 2 neutrons. Tritium is unstable, atomically unstable, which means it decays. And it decays by emitting a beta particle. It decays by emitting beta radiation. Beta radiation, it turns out, are fast, energetic electrons. They're electrons with a lot of kinetic energy. They have enough energy that they can expose photographic film. So after treating the brain with radioactive molecules, their location is determined by placing sections of the brain. They treat the animal with radioactive substances, expose the animal to some stimuli or some situation, sacrifice the animal in a humane way, take out its brain, slice it up thin slices, and then place those sections against unexposed photographic film. The film will be exposed over the radioactive molecules. So in the old days, they actually used chemical film. These days, they put the little sections of the brain on a CCD array, which is more electronic. It's electronic. The principle is the same. We can make certain parts of the brain radioactive and detect them later and see where they are and what their structure is. So as we mentioned before, stereopsis is the ability to see in depth. And in order to produce depth perception, the brain must receive input from both eyes. By using autoradiographic techniques, scientists were able to see how the visual cortex in mammals is organized. And it's organized into these structures called ocular dominance columns. Ocular dominance columns, which are columns of neurons that respond primarily to input from one eye or the other. Now, as we showed, every part of the visual cortex receives input from both eyes. Most of the neurons in visual cortex get input from both eyes, but they'll get input predominantly from one eye or the other. So a neuron that gets input from both eyes, but stronger from the right eye, will be right eye dominant, right? A neuron that gets input from both eyes, but more strongly from the left eye, will be left eye dominant. So the question is, how are these neurons, in terms of their ocular dominance, organized in the cortex spatially? So how do they find out? Well, they injected radioactive proline into one eye. It's taken up by retinal ganglion cells, and transported back to the lateral geniculate nucleus, and from there to the visual cortex, the first station in visual cortex, V1. And only cells receiving input from that eye are labeled with the radioactive proline. Then autoradiographic processing shows the structure of the ocular dominance columns. And what do they look like? Well, what do we mean by columns, first of all? Well, as you recall, the cortex is organized as a sheet. Neural cell bodies in the cortex are organized in a thin layer, like a sheet. That sheet gets crumpled up into the sulci and gyri that we know of in the cortex. In most parts of the cortex, that sheet is actually 6 layers thick. Layer 4, and this is true for the visual system especially, layer 4 is the input layer of the cortex. So axons that come in from the lateral geniculate nucleus are going to contact cortical neurons in layer 4, and those input neurons in layer 4 are going to communicate with other neurons in layers 1, 2, and 3 above them, and layers 5 and 6 below them. So the cortex kind of has this vertical column structure. And different vertical columns are specialized for different functions. What do I mean by different functions? Well, neurons in a particular column might all respond to the same color of light, like red, for example. Or they might all have the same orientation selectivity. They might like bars of light or elongated blobs of some specific orientation. Or they could all have the same ocular dominance. So all of the cells in a column here have right eye dominance. All of the cells in the column here have a left eye dominance. Okay? So now we can ask, because we can visualize them using autoradiographic techniques, how are these ocular dominance columns organized? How are neurons organized spatially in the visual cortex with regard to their ocular dominance? The ocular dominance columns form this zebra-striped pattern, which is pretty amazing. That neurons in the visual cortex will be organized according to their dominance, ocular dominance, in this zebra-striped pattern. This fascinated scientists, from neurobiologists to physicists. And so we've learned a lot about it in the ensuing years. Using a technique called monocular deprivation. I'll explain monocular deprivation in a minute. But in normal animals, the ocular dominance columns for one eye are about the same as the other eye in terms of width, length, and the total area they take up in the cortex. But after monocular deprivation, where you deprive one eye of exposure to the visual environment, the ocular dominance columns become asymmetrical. Nearly all of the space in cortex is devoted to input from the intact eye. And I'll show you pictures of that in a minute. Since the visual cortex is needed for most forms of visual processing in the brain, monocularly deprived animals are blind in the previously shut eye. We'll go into detail on that in the next slide. What is monocular deprivation? In monocular deprivation, you prevent one of the eyes from seeing the world. Here are a couple of monocularly deprived ferrets. You can see that experimenters have put an eye patch over their right eye in each case. And to be cute, they said, oh, don't they look like little pirates? And so they photoshopped this little image of ferrets with pirate hats and swashbuckling swords and all this kind of stuff. But ferrets and kittens and other animals can take their patch off if they want to. So to be sure of monocular deprivation, what most experimentalists do is they take surgical suture and they suture the eyelid closed, which seems kind of gruesome, but it's not as bad as it sounds, because you take the sutures out and the eyelid is normal and the eye is normal and everything's normal, and the suturing is done under anesthetic so the animals don't feel it anyway. But generally speaking, monocular deprivation is accomplished by suturing the eyelid shut. Okay, and so let's say you suture one eye of a kitten shut. Well, here are the normal pattern of ocular dominance columns. The bright areas are where the radioactive proline has ended up after projecting back from the retina through the LGN back into the visual cortex. The dark areas are unexposed film. So in this case, the seeing eye is the one that was injected with proline, and in this case, both eyes have normal vision, and one eye has proline, the other does not, and you can see that the ocular dominance columns are very similar between the two eyes. In B, only the seeing eye has proline. The monocularly deprived eye does not have any radioactivity. And you can see that the visual cortex that is exposed from the seeing eye occupies most of the territory in the visual cortex. Here's a diagram of what's thought to be happening in the development of the visual system on an anatomical level. During embryonic development, the lateral geniculate nucleus sends axonal projections back into the visual cortex. So they're present at birth. But the projections that are present at birth are kind of sparse and spread out. They're sparse but diffuse. They cross all over each other. During exposure to the visual environment, during activity-dependent development, the projections from LGN into cortex become elaborated. They grow out lots of more processes, but they also become more circumscribed. And here you can see that one is becoming circumscribed to one eye, one neuron, and the other to other sets of neurons. This is what they think is happening in monocularly deprived animals, that the input from the deprived eye withers away. The projection from the seeing eye elaborates and expands into the territory that would have been occupied by input from the other eye, but the other eye has been deprived of visual input. We actually understand a lot about this process, and it might look as though inputs from the two eyes are competing for territory in the visual cortex. And that's pretty much what happens. We'll talk about that in a minute. First, I need to introduce the concept of a critical period. The development of ocular dominance columns occurs during a critical period. In kittens, cats were the main animal subjects of these experiments. In kittens, the critical period for development of ocular dominance columns is from 23 to 40 days postpartum, postnatal. That's about week 3 to week 6 postnatal. Suturing the eye shut before or after the period has no effect on the development of ocular dominance columns in cats. So the critical period is very important. The critical period is the period during which ocular dominance columns and other aspects of visual function are going to develop. It doesn't happen before that or after that. But the visual system requires visual input to both eyes during the critical period in order to develop normal visual function. Monkeys also have a critical period, but its duration is a few months rather than a few weeks. Humans also have a critical period for the development of vision, but it's a few years rather than a few months. We'll talk about that in a little while. As I mentioned a few moments ago, ocular dominance columns are formed through competitive interactions among neurons in visual cortex. A projection from LGN to cortex must be activated by input from the external visual environment in order to compete successfully for synaptic sites in cortex. Well, what would happen if you sutured both eyes shut? You deprive both eyes of exposure to the visual world. Well, now one can't outcompete the other, so what happens? Suturing both eyes shut during the critical period renders both eyes equally inactive, so neither has a competitive advantage. Ocular dominance columns in that case appear normal shape and normal symmetry. Inputs from both eyes have about the same amount of territory in visual cortex. This indicates that the competitive advantage of the open eye caused the asymmetry in ocular dominance following monocular deprivation. So by knowing when the critical period for visual development was, and by suturing either one eye or both eyes during that period, scientists were able to show that the formation of ocular dominance columns and really the development of other aspects of visual function involves a competition, literally a competition between inputs from both eyes for synaptic sites in visual cortex. And this is an interesting phenomenon more generally, because competition is a mechanism for creation of structure and function in lots of parts of the brain, and maybe in society too. Here's a photo of our beloved Illini in a scrimmage. So these are members of the Illini team playing football against each other. Why would members of the same team play against each other? They'll set up a little internal competition, and through that competition, they'll develop. They'll develop their skill as football players. They'll develop functionally. A very highly analogous thing is happening in the development of the visual system, in other sensory systems, and maybe in other brain functions. So in these binocularly deprived animals, ocular dominance columns appear normal. Is the visual system normal? Ocular dominance columns appear normal after the critical period in binocularly deprived kittens. However, the neurons have abnormal responses. They respond almost exclusively to input from one eye or the other. There are almost no binocular neurons in binocularly deprived kittens, even though this is the most common type in normal kittens. As I mentioned, we can see in depth because our brain, our visual system, and even specific neurons, individual neurons, can combine inputs from both eyes. They have inputs from both eyes on them, so they can compare the images from two eyes and create the perception of seeing in depth. If you deprive the eyes of vision during the critical period, ocular dominance columns appear normal, but there's no intermingling of input from the two eyes. There aren't any binocular neurons, even though in cats before that, those neurons combined in from both eyes. These animals could not see in depth, but it's worse than that. Binocular deprivation also prevents the development of orientation selectivity. These neurons can't even see little elongated blobs of light at different orientations, and that's the basic visual percept. They can't even see the most fundamental visual percept, so they can't perceive anything. They're essentially blind. These functional abnormalities underlie the cortical blindness that results from visual deprivation during the critical period. Very serious. So let's take a closer look at orientation selectivity. Here again is another diagram of a little section of the cortex. Recall that the cortex is organized as a sheet. All the cell bodies of the neurons are organized in layers in a sheet. Layer 4 is the input layer, and neurons in input 4 project up their column to layers 1, 2, and 3, and down to layers 5 and 6. And as we mentioned, all of the cells in a column have the same functional properties. They might all respond to the same color. They might all have the same ocular dominance. And they will all have the same orientation selectivity. So here we see a diagram of these little oriented bars that I've been talking about. Interestingly, the input layer, layer 4, doesn't have cells that are orientation selective. They just respond to little spots of light. Recall that the retina already detects spots of light. It sends that to the lateral geniculate nucleus of the thalamus, which is essentially just a relay station. It relays those little spots of light into the visual cortex, into layer 4, because layer 4 is the input layer of the visual cortex. But in layer 4 and projecting up and down, and the processing that occurs within the column results in this nice orientation selectivity. The neurons in the lower and higher levels are sophisticated enough to put these little spots together into elongated bars and to respond preferentially to bars of a particular orientation. In this diagram, this is an electrode. It's as though you put an electrode into the brain and we're recording all the neurons in a single column. If you go across the columns, then you can see that neurons in different columns are specialized for bars of different orientations. It's quite interesting that as you go spatially across a column, the orientation selectivity changes gradually. This is a property of visual cortex in general, very, very interesting. It's understood to some extent also. The details are beyond the focus of this class, but it's a very interesting finding nevertheless. The important thing for us is that in the normal visual cortex, the primary visual cortex, V1, neurons have developed the ability to see little elongated blobs of specific orientation. That doesn't happen in an animal if one eye is occluded. Input from that, the brain does not develop the ability to see oriented blobs from that eye. If both eyes are blocked during the critical period, the animal can't see oriented blobs from either eye. It can't build up its visual percept, and it's essentially blind. Here's a nice diagram showing how the visual percept is built up. We start with little spots of light in the retina, go to spots of light in the lateral geniculate nucleus. By the time we get to the first stage, because of activity-dependent, environment-dependent development, the animal can see these elongated blobs of different orientations. At the next level up, V2, these blobs have been put together into little corners, so the animal can detect corners. At higher levels of visual cortex, simple shapes. At higher levels of visual cortex, now moving into the temporal lobes, more complex shapes, and then finally, toward the very tip of the temporal lobes, specificity for faces, and even beyond that, we're still exploring it. So, activity-dependent development that requires activity of the visual system, which requires exposure to the visual environment, is critical for the development of the ability to see. Ocular dominance columns and orientation selective neurons are found in visual cortex area V1, again, not in LGN, not in retina. They appear first in V1. Binocular deprivation will not affect central surround light spot responses in LGN. So if you deprive an animal of vision, its eye will be normal. Its optic nerve will be normal. Its lateral geniculate nucleus will be normal. Those cells will be able to respond to spots of light. What won't be normal are the orientation selective responses in V1, because binocular deprivation will eliminate orientation selective responses in V1, and this will preclude perception of elongated bars, and then everything else built up from them, including corners, shapes, and objects, as we just showed. If you can't see these elongated bars, then you just can't see anything else. And even though your retina is normal and your lateral geniculate nucleus is normal, you won't be able to see anything besides just diffuse light. You won't be able to see. You won't be able to read, recognize faces, drive a car. You'll be effectively blind. So exposure to the environment, visual environment, is absolutely essential. That's why it's very important to make sure that babies can see. When a baby is born, you want to make sure the baby can see. You want to make sure that the baby's eyes are not infected in passing through the birth canal. Often, babies' eyes are treated with antibiotic to prevent infections which could cloud their vision. Sometimes babies are born with congenital cataract. A cataract, as you may know, is a clouding of the lens of the eye. It happens in old people. I'm starting to get cataracts. Very common in people in my age group. But sometimes it happens before birth in children, and their lens is clouded and normal visual input cannot enter the eye. In this case, it's critical to remove that clouded lens and correct the child's vision with eyeglasses. But then at least the child can clearly see the visual environment, and the child's visual system can develop normally. And that visual system development is going to take a long time in human kids. In human children, the combined critical period for normal development of vision, and specifically for development of aspects of vision, including light sensitivity, letter acuity, and global motion, extends from birth to 7 years of age. In cats, it's a few weeks. In monkeys, it's a few months. In humans, it's years. Because we have a lot to learn. Our visual system has to see letters and complicated shapes. It takes a long time for the human visual system to develop. And this is good and bad. It means that you have to have clear vision for that whole period, but maybe not really. because the critical period is so long and the visual system is still in a process of developing. It's still moldable, malleable for the first 7 years. If there's some deprivation, visual deprivation in early childhood, but that deprivation is removed and clear vision is restored, usually the visual system can develop normally in those children. So this process of development is a very obviously active process. It involves neural activity, neural impulses, as carried by waves of ionic current, and it also involves various molecules, transmitters certainly, and also neurotrophic factors, which we talked about before. They're involved in the growth and maintenance of neurons. Two well-known ones are nerve growth factor and brain-derived neurotrophic factor. This is again the picture of the classic experiment showing a cluster of neurons that normally will send out some neural processes. If you treat it with nerve growth factor, the neural processes grow out like crazy. The normal development of the visual system and other systems involves neurotrophic factors. And interesting experiments can be done by removing the competitive advantage of the open eye using neurotrophic factors. Because monocular deprivation will cause axonal projections from the blind eye to wither, but this withering can be reversed by infusion of nerve growth factor. Nerve growth factor works against the competitive advantage of the seeing eye by stabilizing all connections regardless of their activity. It would be like in a basketball game where two teams are fighting very hard against each other, but a robot just picks up basketballs and dumps them into the net on either side at a ferocious rate. And neither team can win, even though one might be better than the other. Neurotrophic factors that influence ocular dominance column formation can include brain-derived neurotrophic factor and the neurotrophins 3, 4, and 5. So this gives us an idea. This gives us a picture of the development of the visual system involving kind of complicated neural and molecular interactions, but which depend ultimately on exposure to the visual environment. Development of language and of social ability also requires exposure to the environment, but in this case, the environment is other people. We'll talk about that in part 2.